It's a pleasure to introduce Dr. Huixia. Let me make a very brief introduction to her. She graduated in chemistry in Zhengzhou University in China. She did also her master in chemistry there, and she did a master thesis on organic synthesis, right?
Yeah.
But then you move it to theory.
Yes.
And she moved to Spain to do the thesis with one of these China Research Council grants to Barcelona, to the Universitat Politécnica with Jordi Martí. And she worked on the molecular mechanism of cancer. And then she returned to China, to Shanghai, where she worked at the School of Pharmacy, working also in simulations of molecular mechanisms of different diseases, including Alzheimer's disease and cancer disease. And after that postdoctoral stay in Shanghai, she returned again to Barcelona with one of these Margarita Salas grants. Maybe you don't know the Margarita Salas program. This program has been discontinued as the Maria Zambrano program. It was an interesting program, but it's not that program. Again, working. This program was a postdoc program in which you do stay in two different places, two different research centers. So it has to be a project coordinated between two different institutions. So it was interesting in the sense that the postdoc with this grant was able to experience two different environments and two different centers, but it's no longer funded by the Spanish government. So we have been talking too much, so it's time to start your talk, and the floor is yours.
Okay, thank you so much for Dr. Jordi Paolo for his very nice, good introduction. And I would like to thank the organizers for this invitation for me too, so I can present my work here. And I'm very looking forward to this talk and to interacting with this fantastic community. So here's a outline of what we will be covering today. First, I will do the introduction and the methodology that I used in my research, followed by the overview of different projects I have been doing in recent years. And then I will showcase the two research projects conducted here and UPC. And then in the end, I will introduce a new research line that allow me to model materials from a much larger scale. And finally, the acknowledgements. So what motivates me? Like using computer simulation in my research, because they are so fundamental for science. When the experiments are impossible to perform, for example, the weather forecast, collision of stars, and when they are so dangerous or they are extremely expensive, and also when the phenomena on scale that are so small, then we need simulation, computer simulation in these cases. Okay. So today's talk is about mainly focused on the protein-protein interactions, DNA, RNA, ligand binding, and nanomaterials. Regarding the size of the systems that I'm interested, so I'm using all atom molecular dynamics. So what is molecular dynamics? In general, the molecular dynamics simulation, you totally rely on the Newton's laws of motion. And each item's motion is governed by some potential energy functions, which is also called force field. And MD simulations can explore the thermodynamics equilibrium states of the system that you are interested in. And it's a computer simulation technique they use to investigate the physical movements of the molecules or atoms, allowing us to explore possible different state of a system, and so we can gain insights into the behavior of materials in your system at atomic or molecular level. And it can help us to predict the behavior of a system even before the experiments are performed, and making it a valuable tool to guide the experiments and also do some hypothesis testing. In general, it's a virtual experiments. So what simulations, MD simulations can simulate? What kind of systems that you can simulate? Here I present some examples: the siRNA, RNA, DNA with the peptides, and peptides with lipid interaction, and doublets of spike protein with different surfaces, or the polymer protein receptor glycan with some virus proteins. You can do a lot and more, of course. So at the beginning of my PhD study, I'm fascinated by the cell membrane barriers. It may seem like very simple to you, but because why I'm interested in this, because of its fundamental to many cellular functions. So that they serve as a selective barrier. They can host membrane proteins, several membrane proteins and channels, transporters, and take part in the signal transduction and cell-cell communication and their interactions. So I have done I have investigated several small molecules interacting with different cell membrane barriers, and our work published in these journals. And in this work, I have combined using the classical MD simulation together with enhanced sampling techniques. And in this work, we have used the two variables of this dihedral angle beside and the distance between the central mass of the melatonin and with the center of the DMPC layer. And here we explore the 2D free energy profile. In this year, together with Professor Jody Maria of UPC, we have constructed the asymmetrical brain cell membrane model. And we have considered several kinds of lipids in this cell model. And the results shows here that the permeation ability of this FDA-approved drug, TMZ, is hugely increased by the raising the temperature in the system. As you can see that with a much lower free energy barrier at the slightly higher temperature, right? And especially the cholesterol and BPGS has been found to play a crucial role in gating the transduction of TMZ entering the brain cells. So here we kindly suggest that the patients who are suffering fever should consider reducing the dose of TMZ, and also they should pay attention to the daily digestion on cholesterol and BPGS as well. So after conducting several projects regarding the membrane barrier systems, we have gained very good understanding regarding the different membrane barriers, like the upper lipid thickness, outer parameter, density profiles, and also we can trace special atom pairs of interactions as well. In the second half of my PhD, my interest shifted to the dynamic of the KRAS4B at the membrane barrier. It's a membrane protein. And why it is so important? And from this panel, you can see that the KRAS4B protein is heavily involved in different cancers: adenocarcinoma, colorectal adenocarcinoma, and the G12D, the KRAS G12D mutant is the highest frequently mutated KRAS mutants. So in these two works, we have explored the conformational space of KRAS4B G12D mutant binding the membrane, and also the several metastates of the KRAS4B G12D mutant has been reported in this work. And in this work, we are reporting, we have designed, because the KRAS4B G12D still remains undruggable. Okay, so there's no effective drugs in the market. So here in this work, we provide a novel idea of designing a lipid-like drug, which serves as a potential binder, which it can bind, not only lock the KRAS4B in its inactive state, and also it can bind the PD delta, which serves as a transporter that it can grasp the KRAS4B and then transport and release it near the cell membrane, and so the KRAS4B can anchor the cell membrane, so it can take into different signal pathways. So in my first postdoc stay in Shanghai, as a principal investigator, I took part in this project of proposing the A-beta oligomer structure, which is very well known for the Alzheimer's disease. And disease caused by the abnormal deposits, mainly caused by the amyloid plaques and tau tangles and so on. And the most frequently studied A-beta forms are the A-beta 40 and A-beta 42. But in recent years, there are several A-beta oligomer has been proposed, but you can see that it's totally not the case for A-beta 42. And it's more prone to aggregation, and it's considered more toxic, okay? So it's playing a central role in the formation of amyloid plaques in this disease, in the brain. So no reported structures of the A-beta 42 oligomer. In the paper published in 2018 that they have captured this through the label-free imaging technique, they have captured this figure. You can see that this is the green thing is the bilayer lipids, and this purple is represents the amyloid plaque. And in the following year, they have this paper they published. They are reporting that extraction of phosphate lipids of molecules from the membrane may lead to the formation of a beta lipid complex. So here several questions has arisen. Okay, how amyloid extract lipids from membrane, and do they form assembly, and are they stable? Do they have any stable structures? So we did our investigation by combining the SSL, AMR, and computational studies by using MD simulation and chemical shifts from SS and MR. In this work, we are proposing several different A-beta 42, A-beta 42 oligomer structures. And there is mono units, dimer units, and trimer units in membrane, and tetramer units in membrane. The manuscript is in preparation due to the difficulty of the experiments. Many thanks to our collaborator, Professor Burchen and Professor Surin of University of Central Florida, and they provided very good SSNMR results for A-beta 42 oligomer, and also from the AFM figures that they obtained from the wildlife experiments, that we can see that in the membrane, we can see clearly there are several A-beta 42 oligomer has been observed here. Okay, so let's dive into the two research projects that I have conducted here and at UBC. Here is the background information. So the T-cell therapies in general involves composites of like extracting T-cells from the patients and do the T-cell proliferation, and then do the T-cell expansion in vitro, and then reject back the enough amount of T-cells back into the patient's body, which is a very promising strategy. But however, obtaining large amounts of therapeutic T-cells in a very short time and also in a cheaper way still remains a challenge. Our collaborator, Dr. Judith and Miguel, which shows up here, they are using 3D hybrid hydrogels to do the T-cell expansion in vitro. And here I want to point it out, here is the standard first aid buffer, the saline PBS buffer is widely used in bioexperiments, and including the 3D hydrogels we are discussing here today. So in 2022, they have published a protocol like how they did in their lab to incubate T-cells, to do the T-cell expansion, right? So first, they will establish the CCL21 loaded 3D heparin hydrogels at the room temperature. And in this first step, the PEG provides structural and mechanical properties. And then the heparin, the negatively charged heparin polymer, will serve as anchoring point for the positively charged CCL21. After equilibrating the complex of this 3D matrix, and then they will introduce the T cells into this hydrogel at the human body temperature. So this is a scan. Our collaborators are interested in knowing what is going on from the atomic level, right? So they can increase the T cell proliferation in their experiments. So first, we had to construct the system of heparin CCL21 in PBS, solvated in PBS buffer. And we run the simulation, and in the end of during the simulation, we have found unexpectedly we found the phosphate aggregation in our system. So here, several questions raised. Okay, let's take a look at this aggregation. So does this phosphate aggregation affect the heparin CCL21 interaction? And does it play a role in the folding process of T cell expansion? Will it affect influence the T cells, and then under which condition the formation of this phosphate aggregation will appear? So this makes us very curious. So in order to get a better understanding of the role played by these phosphate ions, so we removed the phosphate ions from the system. We only consider sodium chloride of the physiological concentration. We run the simulation, and then we get a well-established complex of the CCL21 and the heparin solvated in the sodium chloride solution. And here we calculated the contact area between the CCL21 and heparin in these two systems. We have found that with no phosphate aggregation occurs, when there's no phosphate aggregation occurs, that we have observed the increased contact area between the heparin and CCL21, right? So it indicates a stronger interaction between them. So you may want to say, okay, maybe you can just remove the phosphate from your system, from your wild lab experiments. Maybe you will get a better T cell results in your experiments. But sadly, no. When we get the samples of T cells from the patients from the hospital, the T cells are solvated and preserved in the PBS buffer. So in the second process of the T cell infusion into this hydrogel, the PBS will reinfuse back into the 3D hydrogel. So how about we establish the CCL21 and heparin first? And then we will see, let's investigate, will this later on addition of the PBS will affect the stability of CCL21 and heparin complex. So we take the final established heparin CCL21 complex without the phosphate, right? And then we put PBS back to this system and then run simulation. And in the end, you can see that along with the refusion of the PBS, the phosphate assembly appears again in this system, but seems like the orientation of the CCL21 and the contacts between them seems they stay the same. Is it true? So we calculate the contact area. And you can see that the phosphate assembly along with the PBS refusion into the hydrogels the contact area between heparin and CCL21 doesn't decrease compared with the protocol one, which is good news for us. And then by only looking at the contact area is not a very scientific way. No, you can say like, okay, this higher binding affinity. So we evaluated the binding affinity. We calculated the binding affinity between the CCL21 and heparin in these three systems. As you can see that in these systems, we have all observed a much stronger binding affinity between the CCL21 and heparin. So here we propose the protocol, a new protocol, how to prepare the 3D hydrogels, right? First, you equilibrate the CCL21 loaded hydrogel without the phosphate, and then you can include the PBS back along with the T cell infusion. So what's the impact of this hydrogel on T cell expansion? We only investigated the interactions between CCL21 and the heparin. How about the interactions between the CCL21 with the T cells with the CCR7? What is CCR7? Let's take a closer look. Okay, so in the second phase, when we introduce the T cell into the 3D hydrogels, and the CCL21 can relocate T cells into the 3D hydrogels. How? By interacting with the CCR7, which is the CCL21's receptor, and it is a membrane protein of the T cell. And we also evaluated this binding affinity between the CCR7 and with this complex in the three systems. And you can see that, okay, we have observed a much stronger interaction between the CCR7 and CCL21, indicating a better T cell proliferation in experiments. So our collaborators, they are very nice. They go through a lot of difficulty, did the experiments, and the wildlife results are in good agreements with our prediction here. So take-home message: if present initial, the common use of PBS can compromise the stability of the heparin CCL21 interactions. And so in this work, we are introducing a novel protocol for 3D hydrogel preparation, which enhances the proliferation of T cells in wildlife experiments. Okay, here again, we take a closer look at the first phase aggregation, right? So we wonder under which condition of the formation of the first phase aggregation will appear. So first we run a simulation of the pure PBS simulation box, and then in other simulation box we included the heparin of different lengths of heparin and even the PEG that is considered in their hydrogel, right? And so we can see that with the addition of PEG and different lengths of heparin, we have found at the end of this simulation, we have found a single cluster of all phosphate plus sodium cations, a single cluster here. But PBS itself is very stable. And PEG and heparin molecules can trigger the first phase aggregation. And we also consider different heparin, different charges of heparin, and all the charge density of the heparin may influence the speed of the aggregation, but in the end, we all get a very single cluster in the end. So what triggers this formation of the aggregation? And we have calculated the evaluation of number of water molecules around all phosphate ions and sodium cations. And you can see that by comparing the difference of water molecules in their water shell, we can see that at the end of the simulation, in 3 cases, around 170 water molecules has been released from their water shell to the water bulk. So we propose here is the entropically driven phosphate aggregation in PBS is thermodynamically favorable through this entropy composition of the water molecules. And we also investigate the effect of the charge density of the counter ions. And in these cases, we can see that the charge density of the counter ions also play a role. Like the phosphate assembly only appears in the cases of lithium and sodium, and not in the potassium and rubidium. So which is also very interesting. Thanks to Miguel again, like our collaborators and also Dr. Judith, they did the cryo-time experiments. And as you can, by comparing that the nanoparticles, they can see these small little circles that appear in the samples of the PBS plus the heparin, right? But you can see that in PBS it's quite clean. So it's confirmed, like in the PBS it's stable and there's no aggregates. And also they double checked by doing the DLS experiments, and you can see that here I highlighted here is like the increased size of particles in the PBS plus heparin and in the system compared to the single PBS reconfirms the existence of the phosphate assembly here. So the phosphate aggregation is primarily driven by entropic forces from the solvation water, and in potential impact of phosphate assembly in PBS on certain bio-experiments shall merit significant attention. Materials that you don't care may play a role in your experiments. And now I will introduce a new research line that I newly developed at ICMAB and UPC. So for now, you can see that I have done all my research using small molecules, proteins, and sometimes I do coarse-grained empty simulation, and all the projects are conducted at the atomic or bit level, right? So can we simulate the cell models, different cell models, or even bigger, can we simulate tissue or a whole organ? The answer is yes. Oh, sorry. Oh. Can anyone help me? Okay. Sorry. So the CC3D, CompuCell 3D, CCL3D is a computational modeling platform that developed by Professor James Gleiser's team at Indiana University, Bloomington. And it is designed to simulate the biological processes. And here I show the simple CC3D models of the tissue growth, or I don't understand because I'm not a biologist. Here I show a very nice simulation of the tumor growth and using the CC3D, and you can see along with the simulation time, and the tumor starts to grow bigger and bigger. And it is also affecting the nearby tissues. Okay, the CC3D uses a Monte Carlo metropolis algorithm. And in order to build a good model, a good cell model, you need to take care of a lot of different parameters, such as contact energy between different cells, that their target volume, when to decide they will divide, or how much of the volume they will end up growing, right? And a surface tension between different cells. This project is initially developed in a hackathon project in 2024 August, and a pre-doc, Mr. Omar, has joined this project. So what motivates me to do this project, which is the construction of a multi-scale model for investigation of the impact of CCR signaling on the T cell growth or proliferation, is because this impact of CCR7 signaling on T cell proliferation is not clear. So ultimately, I want to build a model that consider 4 different cells, like the heparin polymer cells, the CCL21 cell, a whole T cell, and maybe several CCR7 receptor. And that is still like going on this project. But the preliminary results here is, in order to get the contact energy, which is the very essential parameter to build a good cell model, that I explored the full-length CCR7 system. And after getting the representative structure of each cluster, which is the C2, is the most stable according to the density of the structures. So I use the C2. In the next step, I will use C2. I will estimate the CR7, the contact energy between CR7 and CCL21 to get a proper value of the contact energy. So here is the initial number of CCL21, which is the blue dots you can see, and it set the initial concentration of the CCL21 is 1,000. And in the end, after 10,000 Monte Carlo steps, and you can see that in the simulation box that T cell is everywhere, which is not the fact, right? They are not so easy to grow in your lab. And I also did the investigation on the effect of a concentration of CCL21, and you can see we get different results. And I also checked the simulation by deleting the consumed CCL7, and here I present the number of T cells along with the simulation time and the number of CCL7s in the current simulation box. So this is still an ongoing project, so additional results is pending. Acknowledgements. Okay, the simulations mainly performed using the Moneynaughton 4, Moneynaughton 5, and also CESCA FI3. And many thanks to our fundings, and I really appreciate it, especially supported by Margareta Salas grant funded by the European Union Next Generation. And our beautiful partners, thank you so much. Without you, this project cannot be done. Thank you for time. I open floors for any possible questions. Thank you. I did it too quickly.
So the microphone is not working, so we'll have the questions without the microphone. Any questions, comments?
Yes.
Maybe when we start with the two simple comments.
Yeah.
First, I have enjoyed very much your talk.
Thank you.
Really interesting, really very well explained all your trajectory in the recent years.
Thank you.
And it was clear. And I am curious about the project, this project you are starting now.
Mhm.
Two aspects. One of them is the use of AlphaFold. Because recently it has been given the Chemistry Nobel Prize to one of the authors of the AlphaFold, I think.
Mhm.
So do you think this will be a reason for less problems about using AlphaFold in the next future?
I hope so. I hope so. And a lot of scientists that don't believe right now, they hugely don't believe the structures like predicted by AlphaFold 2, right? When we submit a paper, in general, if you mention that your model, your protein model is predicted by AlphaFold 2, and then they simply says, we don't buy it, or give you a lot of troubles. But in this system, because the transmembrane domain of the CCR7 has been published using cryo-EM, and the only part we are using AlphaFold 2 to predict is only this small domain, this N terminal and the C terminal. So that is mainly what we are doing now to explore the possible and the most stable active conformation of the C N terminal and the C terminal. As you can see, it's majorly here.
Okay, and the other thing is about this project. What is the time and length scale of the simulations you do to simulate the growth of the cells?
In this, we can see it here clearly. Like here, in general, it's like 20. It's up to you. You make the rules of your cell models.
This is quite far away from the atom level.
Yes, it's quite far away. They're using a different algorithm.
The growth of a tumor maybe is about days or months or years.
Yes, but in your simulation, you can fast them up. You can say, okay, I define the growth of the tumor to a certain volume to be like 20,000 Monte Carlo steps. And because in the end, you will need the experimental data to refine your model. You can propose a preliminary model at the beginning, and then you feed in the experimental data to refine it.
They use Monte Carlo. It's no real timing dynamics.
I think so. They are using the Monte Carlo metropolis algorithm that I just dive into this project. Mm-hmm.
Start initial final state, sample intermediate states, and you do a lot of sampling for each state. Okay. Thank you very much.
You're welcome. Yes.
I have 3 questions.
Yeah, please.
Is that okay? Okay.
Please.
The first one was really related to the previous question you mentioned AlphaFold.
Oh, yeah.
And you said you were using it for that part of the proteins hanging out into solution. So, in general, does AlphaFold predict well protein structure when it's in solution? Because, as I understand it, AlphaFold takes all of its learning in solid state.
Yeah, they mainly learn from the already existed structure already published in protein data bank. No, that is why the molecular dynamics are so important, because in general cryo time they do the experiments. They do experiments at the 4 Celsius degree. Celsius, sorry, I suddenly cannot right. They do the experiments at 4 sorry, I forget again. Thank you. Celsius temperature is quite low. So in the room temperature or in the human body temperature, the dynamic of protein should be dynamic. So that is why we are using AMD simulation to mimic this physiological condition to see that is why we are getting so many metastable states of the CCR7, which in the cryo time in general we only get 1. or likely to. Yeah.
It's about your phosphate aggregation.
Mm-hmm. Yeah. Yeah.
The picture.
Yeah.
So in your phosphate aggregate, that really surprised me. So I always thought phosphate has been very solvated. As your solvent, you're not using an atomistic solvent, it's just a water dielectric field. Or is it really automated?
So you have explicit water, that's the question.
Yeah, I have explicit water. And actually we have tried to use implicit water. And the amazing thing is the implicit water, have some additional slides. You can see here that we are using this phosphate aggregation is quite stable if you are considering the explicit water molecules, that's water molecule models. But if you change using implicit water, and it is directly dissolved. So it's a water molecules, the water shell, that is the key reason why this phosphate aggregation stays stable.
So I assume that within these phosphate and something like
yeah. Okay. Yes. So we tested the charge density. I believe this is the slide you're looking for.
And last question.
Please.
It's a bit more philosophical. So you mentioned about mechanical properties and dynamics of things. But the big difference between a lot of these systems is knowing kind of what kind of state you're talking about, I suppose. So especially when you talk about membranes, people talk about solid or liquid membranes and that kind of thing. So do your models give any information about that when you look at your molecular dynamics?
Yes.
Can you kind of tell the difference between solid or liquid membranes?
Yes, exactly. That's a very good question. Let me go back to the membrane barriers. Okay. Here. By calculating the other parameter, and if the value of the other parameter is too high, and then we can say that it's in the crystal ordered state phase of the membrane barrier. If it is around 0.2, and you can say that they are in the around the liquid disordered phase of the membrane barrier. Yeah.
When you do your modeling, can you see areas which are more solid?
Exactly. When you do the simulation, if it is like gel phase, it's more like tilted in the same direction. Also, you can see clearly in your simulation by looking at the trajectory. Yeah. Okay. My pleasure. Any questions?
More questions? Okay, so if we have no more questions, we can thank the speaker again and go.
Thank you so much.